All right, so this is our last lab working with Bio 1. We're going to cover the senses, and we'll focus on two special senses, hearing, which includes vestibular and vision. So first we have to overview what we mean by sensory receptor. We've covered that through our arc through the nervous system. We have sensory information coming in, processing center, motor information coming out. To receive the sensory information, we have to have what's called a receptor. The receptor's job is to convert something in the environment. The things in the environment are called stimuli, like that crashing sound, and then it'll convert it into an impulse, which will transduce it through to the brain and tell us something had happened. External receptors sense things that are outside of our body. These are the senses that we know about: taste, smell, sight, hearing, vision, and the sense of balance and equilibrium. This is a list of everything we'll cover today. I don't need you to know that specifically, but this tells us the type of receptor we have, what they're stimulated by, the category they fit under, what we call it, and then the name of the organ. So this is a great summary of what we're going to cover, but in general we're going to say, are we sensing chemicals, which is smell and taste? Are we sensing light, which is only the eye? That's called a photoreceptor. Are we sensing movement, which could be a mechanoreceptor? Or are we sensing touch, which is not a special receptor, but that's coming up soon. So the interones are inside our body. We have things called baroreceptors, which, like a barometer, measure pressure. These are inside of our blood vessels and telling us how much pressure they're under to help set the negative feedback to stop that. We also have osmoreceptors for osmosis. Does that make sense for water salt? Chemo is looking for pH in this case, so the chemicals, specifically the hydrogen ion, telling us what our pH of our blood is. And then this is all part of that negative feedback homeostasis that we've spent so much time on in this course. The specific chemos are taste, smell, and then the blood pH. Nociceptor is another type called a pain receptor. Noci means pain in this case, and what we're looking for is damaged tissue releasing chemicals saying ow. When they say ow, we're alerted to pain, and that's one of the sensations that's very hard for us to ignore. That one is difficult for us to tune out. Photo responds to light, back of the retina is where we do that. Mechano is a mechanical force. Hearing or sound waves, balance is tipping. Pressure or stretch or pull on muscles, tendons, skin. And then thermo is temperature changes, and our hypothalamus up in the base of our brain is dealing with that. Our skin also, of course, detects temperature. We know how we're feeling by the outside environment. So that's stimulus. Sensation is what happens in the brain when the stimulus has caused a nerve impulse to rise up the afferent pathway and show up in the parietal lobe if it's touch, occipital lobe if it's vision, and as so on. Remember, everybody goes to the thalamus, except for smell. When they arrive in the cerebral cortex, whichever portion of it, we're going to have ourselves a feeling. That feeling is what we call sensation. And when we put them all together, we have integration, which is the summing of signals. So that is a variety of things that we put together to determine what's going on in our environment: touch, and taste, and smell, and sight, and hearing all go together. That's an image of it. And if you recall, our little sensory neuron is going to have his cell body off to the side just before he enters the central nervous system. He's going to be in a ganglia just outside the spinal cord, which you see there. So he sits right about there. He's going to arrive in the back, go on up to the brain. What starts the whole thing is the stimulus. The receptor receives it, and that sends the nerve impulse. Adaptation is the sense that we hear the signal often enough in the brain and we say that's nothing I have to worry about right now, and we'll adapt to it. So there's a humming of an air conditioner, there's uncomfortable seats you're sitting on, there's temperature of the room, we get used to it. We're like, that's okay, I can tolerate this. If something new happens, you'll be alerted to it. Adaptation is less impulses coming in, and it gives us a chance to get used to our environment without always being overstimulated. Some ideas of attention deficit would be lower adaptation ability, so that we're always stimulated by our environment and we're having a hard time finding what's a salient focus point. We also know that pain does not adapt well. So as I mentioned, pain is a signal that we're not going to have an easy time turning it off. Soma, we've used that word before to mean body. So our somatic senses are in our skin and our muscles, our joints, and the viscera, or the guts of the body. The three types here can be proprioceptor. That word will always refer to your body position in space, so it's what's happening in your joints, your muscles, and tendons, and it'll say, I'm standing up right now, and I'm upright, or whatever your body is doing. Cutaneous means skin, so it's our receptors in our skin that tell us what's happening to us. And then of course pain receptors. Pain receptors are everywhere in our body. We don't want any part of our body that we can't be alerted if there's something going wrong. All of these go up the spinal cord to the primary somatosensory cortex, right here in the postcentral gyrus, and then it'll tell us where that body part is based on the little homunculus we saw that shows. I don't have a brain in front of me, but in the center of the lateral sulcus, you'll see the feet moving up to the knees and going around to the trunk, and then all hands and all face, if you remember that from last time. As I mentioned, proprioceptors are dealing with our body position, so they include the tone and equilibrium and posture that help us use our muscles. And there are little spindles inside our fibers, like little springs that will pull or relax depending on what our muscle's doing. And that will send a nerve impulse that you're stretching or you're loose. One of the fancy ones in a tendon is called a Golgi tendon organ. Probably the same guy who got the Golgi apparatus named after him. He is the same guy. He got two things named after him. It was very important. An Italian scientist who taught us how to stain and find things, and I'll move on. But anyway, they work together to tell us our muscle tone. So the knee-jerk reflex that we use when you're having your knee tapped and you see what happens, that is testing this to see if we get a response from a specific input. So there it is. So the quadricep muscle is what we're stretching. We hyperstretch that by tapping it on the kneecap, which will make the muscles stretch. Goes through the spinal cord, comes right back out again, and makes that muscle contract. The proprioceptor is what told us that needs to happen. The skin, here's some other named things, but these you don't have to remember. There's a whole bunch of names here that are going to deal with how we process our sensation in our skin coming in. So we have, and I don't need you to memorize this, just know that these are all cutaneous receptor types, and each possible sensation we have has a very specific receptor for it. So if we're touched, we can feel pain, heat, cold to the free nerve endings, the Merkel discs, the Krauss end bulbs that are a little deeper, you'll see. Our root hair plexus says if something brushes along our hair, like a tiny bug on our skin, we may not feel it on the skin if it's too light, but if it tips a hair, we'll feel it down there. We've got Ruffini endings, which will feel pressure, Pacinian corpuscles also for pressure, and the myosin corpuscles for touch. So tons of little names. AP1 will let you play with those further. Now we move into some special senses. Chemoreceptors for taste and smell both work the same, and they both deal with a chemical dissolved in a solution. So liquid or in the air, a chemical in the air that we smell. The chemical is the stimulus. It binds to the receptor part, and that triggers the brain to say what's going on here. We have viscous stimuli in the olfactory cells, and there are little hairs that dangle that say, here comes a chemical that's floated across the room or in front of me if it's my dinner. And then taste, pH receptors have to be touched directly by the chemical. So in our mouth, we have taste receptors along the tongue that will trigger a sensation going in the brain that says that's what this tastes like. Taste itself is usually combined both smell and taste. It's a sense that we enrich through both experiences. The taste buds are where the little bumps on the tongue are what their function is. They're called papillae. We've seen that word before. It means little finger-like bumps. And we also have them in the hard palate, which is the roof of the mouth, the back of the throat and pharynx, and along the epiglottis. But the tongue is where we're mostly used to seeing them. The 5 types, go over the 5 types of flavors we can have. And again, flavor is a mix of these, and smell adds a lot to our experience. But sweet, sour, salty, and bitter, those are the only ones that were around when I was learning it years ago. We also used to think the specific parts of the tongue would only do specific tastes, but we now know it's more diverse. And then a new one was added over the years, umami, which is a savory, meaty taste. Oh, there's a tongue looking at you. There's its little bulbs up close. And then if you zoom in on one taste bud, it looks kind of like an onion to me. And there they are receiving in the pore a chemical. The little cell is going to send a signal up this nerve ending, which is a dendrite, and then go right up to the brain and say, I might taste something. That goes through what we call cranial nerve, and we looked at those last time, but that's for AMP1 as well. And that describes it right there. The parietal lobe has a taste cortex. Remember, this part of our parietal lobe right around here is all tongue and face, and then the tongue portion, we have a taste cortex, which is going to interpret up as what we just tasted. If you remember, sensation doesn't occur until it hits the brain. The stimulus is the food you're chewing. The sensation is in the brain. And as this points out, as I mentioned, most of what we perceive as taste is really smell. Your nose is stuffed, you don't taste food as well as you did. Olfactory cells are in the roof of the nasal cavity, and they're dangling down, I did this picture before, but little nerve endings dangling through the cribriform plate of your ethmoid bone. And when scents enter through the nasal passage, they will stimulate those neurons, which are called olfactory cilia, little hairs, and they will send specific signals up to the brain to identify what you just smelled. Remember on the sheep brain we did last week, the olfactory bulbs were gigantic compared to this brain, where ours are farther back up in here, so we don't have nearly as much brain processing as animals do for scent, but it is still very important sense for us, and it does trigger direct emotional memories because it does not have to go through the thalamus like everything else. And there's those little bulb bud smells coming up from the rose. Here's our dendrite endings hanging through, which we call the cilia. Hits the bulb and goes straight back to the brain. And here we see it zoomed in. And it's just the end of one neuron talking to the next to say, hey, let's send a message up to the olfactory tract. The cortex is part of the temporal lobe, and it goes directly into the limbic system, which is our emotional base. We talked about it in lecture Monday about the amygdala and the hippocampus, all those parts that are really emotionally based. So scent tends to give us a real strong emotional memory, like grandma's cookies or a favorite perfume on a loved one. And unfortunately, we do lose olfactory cells as we age. This is what we're going to be dissecting tonight. So I'll show it here, and then we'll take a model apart, and then we'll take the cow eye apart to see the same structures. Our eye works like a camera, and that is less easy for us to understand than when we had analog cameras. But if you're familiar with an analog camera, the old-fashioned, you'll recognize some of the parts as we name them. So the eyeball itself, all you see is about this much of it, and it is surrounded by a hard tissue type called a sclera. The sclera is the outer coat, and the front part of the sclera goes clear, and we call that the cornea. And all the rest of it is hard tissue all the way around, so it's a big ball, thus the eyeball. The choroid is a middle layer, so underneath that, you see a layer of vessels, and that is called the choroid, your little blood vessel layer. And then beneath that, we have your retina, or on top of it, I should say. So if we're going out to in, it's the last one, but if we're looking in to out, it's what you see first. So choroid's in the middle, retina lines the inside, and sclera on the out. The choroid layer also builds this structure here, that we call the iris. The iris is an aperture, like in a camera, that would regulate the size of the hole. So the hole is called the pupil, and if my iris is going to constrict, I have a little tiny black dot that looks like my eye, and I can let in very little light, and if it dilates, I let in a lot of light, you see a big black circle. Behind it is a lens. And the lens is part of, again, we have another layer here called the ciliary body, and this will hold the lens. Get the cornea out of here, look at my lens, grab the wrong point, there he is. The lens will sit right here, and he can be changed in its width by how this muscle interacts with it. So it's a ciliary muscle will make the lens go flatter or thicker, depending on whether they're looking up close or far away. And this is where we run into problems needing bifocals, because our muscle weakens over time. So here's the image. You'll see the sclera all the way around the outside. The next layer in, which is full of blood vessels, is called the choroid. Choroid means like a net, so it's a webbing of vessels. And then this inner lining, very, very thin and all neuron endings. This is also considered part of the brain, really. The retina is brain tissue. Is your inner coat, which would be the film of the camera if we're going back to the old days. And that's where you see the blood vessels through it because it's so thin. But the blood vessels are in the lower layer. The retina will go back and form this optic nerve, which is our second cranial nerve. We saw it in the brain that we dissected as an X because it crosses right behind the eyeball just before the pituitary. And then if we follow the sclera around, it'll bulge out in front, which was this piece I just held up, which is our cornea. And there's spaces in between, we'll name it a bit. It goes back around and hardens again. We don't want the cornea, although it's as tough as the sclera, we don't want it solid. We need it clear, or we couldn't see. So it changes reaches from here to here, and there are no blood vessels in the cornea, and that's why we can do surgeries easily. There's no blood here. Just put it right back together. I should say easily, but they're not. The iris, as you'll see, is the colored area. Iris means rainbow, so this is the colored part of your eye, and it's just a circular muscle, as is the ciliary muscle behind it. The ciliary muscle attaches to the lens with little strings called suspensor ligaments, and they look like trampoline, kind of. And if they stretch, the lens will flatten, and if they pull in, the lens will get thicker. Meanwhile, the iris in front of it is determined purely by light, and that's a reflex. So if the light is bright, the reflex will say constrict. If the light is dim, the reflex will say dilate. When you look into your eye, you're going to see black because there's no light in there. So the pupil appears black. But if we go back to the old analog cameras, and I know you have family photos like this, somebody takes a picture with a flash, and everybody's eyes are bright red. They're like caught off guard. Then we eventually developed the camera that does a quick flash and then the real flash. The quick flash makes you constrict, and then your eyes don't appear all red. Why do they appear red? Because the light shines in, you see the blood vessels. And that's the blood vessels bounce back as you see literally the blood in the back of your eye. Now, on an animal like the cow that we'll take apart, and the cat, the dog, and the deer, you may notice, I don't know if you call it cow eyes at night, but most of us lately around this area will see deer ears and deer eyes at night. You see their ears too. Their eyes will shine back at you like a goldish color, greenish. The cats, dogs, same thing at night. They look right at you and their eyes come back. Their retina is coated differently than ours, and what bounces in, the light bounces back, shows that color because they're not constricting against it, they're just looking right at you. And what you see bounce back is that special coating inside. So when we open the cow eye, you see he has a shiny coating in there. It's opalescent. Like ours is not like that. Because we're not designed to see in the dark. Other areas to mention? What's that?
The fovea centralis, is that also the foveal dip right there?
Yep. So that is your macula, which is your sweet spot.
So I work in ophthalmology almost.
You know all about that thing. Yeah. So when you look in the ophthalmology photo, you're going to see two things. You're going to see an area that's kind of orangish, a little circle, and just to the medial aspect of it, you're going to see a bunch of vessels coming out. And the nerve and the vessels are what we call the blind spot, because there is no ability to see right there where my little pointer is. You can't see there because there's no retina. You're designed to have light come in through the pupil, hit the lens will focus it, and put a smack dab on that fovea centralis or macula. That gives you the best vision possible. Because of what's in here, little guys we call cones. Out here we get more rods, which are nighttime vision. So the animals are full of rods compared to cones, and we have far more cones because we're designed for color vision and bright light. Now, if you've heard of macular degeneration, that goes away through a disease process, and you end up with two blank spots in the middle of your vision.
It could also, instead of being inverted, it can go upwards like
Yes.
that we see, and then there's a lot of fluid.
If they swell out, right?
There's the dry empty, and
Yep.
then there's also the
They're dry and wet, right? So the other thing that you'll see in the eye, and again, you're the expert because you work in this, but I've learned over the years, if you have heard of glaucoma, you have too much pressure in the eye, that's why they puff you, and you're blink. But if the pressure's too high in here, it'll actually peel the retina away at the corners. So if the retina peels, now you lose vision here, but you're not going to notice it for a while until it peels. So you get to about here, and now it's too late. You have tunnel vision. You've lost your periphery.
I know somebody got that in like their 20s.
Yes, and it's rare, but it's a
My
serious.
best friend is 33 inches.
And she has it. Yeah, that's a serious, because you detach the retina. You can also get detached retina from head injury, shaking, or severe hit below the head. If the retina is not attached, you're not seeing.
Wow, glaucoma suspect.
The glaucoma is that pushing up in there. The other guy that will happen is if we get this thing cloudy, we'll call him a cataract, and they can just replace that now. That's what I mentioned about popping the cornea.
My cornea is
And you have a cornea issue. Did you get scarring in it from something flying at you at work? You always have a work injury.
shingles.
Shingles.
Oh, yes,
There's another one.
So
Yeah.
my right eye.
So he's scarred?
Yeah.
He's a scarred cornea.
Yeah.
Yeah. So that's like a cracked windshield or a scraped windshield. You're not seeing through it.
Yeah,
there.
it's just blurry.
Yep. And then with light, it would make it worse. When the light is green. Yes, sir.
How come, like they test you, or like they check the inside of your eyes for like diabetes or blood vessel
Yeah.
clots? Why does that
So
happen?
you're looking in here. anything at the distal extremity of your body, fingers, toes, and the eye is one of them, is at risk in diabetes for losing its, because we're not giving the blood sugar to the actual, rephrase that, the sugars in the blood is not getting to the cells that need it. So we start with a spotty damage here. Some cells over here will die, some over here will die, and a diabetic retinopathy is retinal disease that is scattered throughout. So you have different spots in your eyes, in your vision.
Yeah, I
So
worked
it's a
with a
biggie
guy.
to check out.
I worked with a guy who went blind for like
Yeah.
a few weeks because he had diabetes.
Yeah, that's a problem.
Macular edema.
Yeah, and you know, any swelling in here is gonna make it worse. I know you, I joked that maybe something terrible happened to your eye in Israel. Did you get bleach in the eye or something? And lie.
Sodium hydroxide.
So, yes, I remember that stuff. So, your protective coating here, right down here, is called the conjunctiva. I didn't think we put it on here, but it dips up right there, turns around, covers the front, and turns around again and seals it. So if you wear a contact lens, you know that if you put the lens right on this thing,